Thank you so much, Petra. It's just a delight to be here and to celebrate this wonderful 50th anniversary. And I think, as has been said before by at least one other speaker, Rolf, 50 more wonderful years is what we all hope to see unfolding for the DKFZ. Z. Sorry, I'm in Europe. I'm going to begin with thanking people in my lab. Could I have the forward? Could I have the next slide? I'm doing it right. Yes, yeah. Because, of course, none of this work is done in isolation. And I'm just taking the tip of the iceberg here in terms of our trajectory in telomere research and just pointing out people with whom I've been more recently collaborating and the wonderful folks in my lab, because I'm going to focus mostly on fairly up-to-date experimental research. And these people, particularly those highlighted in white, will have contributed most to the work that I'll be telling you about today. And we've had fabulous collaborators at UCSF, a psychologist, Alyssa Eppel, human geneticist Neil Risch, cancer genomics and omics expert Joe Gray, and Kathy Schaefer at Kaiser Permanente, and you'll hear what that's about. And I do want to emphasize what a great joy it's been to work with so many terrific people, not only these most recent ones, but over the years. Now, the topic is telomeres, of course, and the way I'm going to organize this talk is I'm going to give a kind of fairly overview where it's shaking out that telomeres are working out in humans, what's emerged. And I'll emphasize particularly the very interesting and complex world of cancers, very much in the plural cancers, which, as you know, is hundreds, at least a couple hundred of different distinguishable kinds of diseases. And then I want to move a little bit away from the telomere itself. Telomeres, of course, are at the ends of chromosomes, and they're maintained by a very complicatedly regulated set of machineries. And I want to talk a little bit about a couple of things we've been finding about components of telomerase, the machinery that's maintaining telomeres, and how it's impacting, we think, on other processes in cells. So the point for the junior people here, the young people looking into, is this all over yet? The answer is no. There's an awful lot we have to learn still. But I'll begin with really a much more overview, sort of where it's shaking down to in humans. So telomeres, of course, cap the chromosome ends, and cytologically they were known for many years, but not until the 70s, as Petra mentioned, did we find the molecular basis for what was really going on. And first of all, there's a scaffold of very simple repeated DNA sequences at the ends of chromosomes, and those serve as the all-important nucleation and landing site for proteins that are very well studied in aggregate, and there's a lot known about the individual proteins that protect and make a sheath using this DNA as a scaffold. Now, the problem with this DNA scaffold is that it's not that stable, and the problem is it's prone to shortening, particularly because this DNA underneath it is prone to shortening. And when that happens, if the telomere gets too short, there's a very clear set of cellular responses, and all cells that have this maintenance mechanism of telomeres at their ends of chromosomes show this general kinds of things, but they've been particularly well worked out in human and mouse systems. And basically, what happens is the cell ceases to divide, sends a strong signal. It's a persistent DNA damage signal when this end is no longer protected. But it's very interesting because there are other sequelae that are happening. Changes in the whole program of the cells occur, including losses of mitochondrial functions, and of course, this unstable end can, of course, engage in various transactions that will end up with genomic instability, which in the face of other settings like loss of checkpoint functions could have the theoretical ability to promote cancer. And as I'll mention, in humans, there's at least reason for some cancers that that's the case. So telomerase attempts to save the day, and so this enzyme just shown in this very simple cartoon with just the core components and not a number of other known biochemical components has the property of using a small sequence within its much larger, and you'll hear more about this RNA component, and that is used to template the addition of telomeric repeats. And those sequences are the ones that then are important for attracting the sequence-specific binding proteins, and they're proteins that make this sheath. And I've just given you a couple of names for the human ones, which I'll be referring to later on. Now, there are other components as well. So this interaction with the DNA, which of course is not bare, there's proteins on this, this is exquisitely regulated by enormous numbers of controls. I like to use the example of baker's yeast, S. cerevisiae. Now it's been counted over 400 different genes will influence the balance of telomerase action and other degradative processes. So this is under enormous numbers of controls. And in humans, not only are all the proteins, and which will govern interactions with telomerase and other factors, degradative or otherwise, not only are those proteins highly regulated, but telomerase is regulated by a bewildering number of factors, and I've just listed a few of the kinds of things that have been shown to regulate telomerase. And I will actually talk later about splicing control, particularly as relates to the TERT. So in other words, what we've got is an extremely nuanced situation where the general theme, except for things like certain stem cells and for certain activated cells and germ cells, the general theme is humans are really trying to keep telomerase levels low, making our lives difficult. But so that's the context. And so the simple situation we found in the model organisms, which was predicted from incomplete DNA replication, and which if you had insufficient telomerase, you'd predict this would happen. And that does happen experimentally in single-celled model organisms like tetrahymena, where we found telomerase. But that's much more nuanced. As I said, in human cells, there is severe consequences, not just senescence, which initially referred partly to the cessation of cell division, but also a set of functions of the cell which are often very abnormal, and it happens after a delay. And it also, as I mentioned, has, including among it, mitochondrial malfunction, but very importantly, pro-inflammatory and tumorigenic factors have been found. Judy Campisi really pioneered some of this work, showing that these cells can have now distant effects as well. So not only do their self-renewal fail, but they're not able to be normal contributors to their environment. So they're bad news, is the bottom line. But telomerase can keep things balanced, and that's what happens in the, essentially immortal organisms like tetrahymena or budding yeast. Immortal are defined in a certain way in a very biologically limited context. But the point is telomerase can keep the telomeres maintained enough in the face of all the various degradative processes, including incomplete DNA replication, so cells can keep on going. And this is a real characteristic of, if you total up human tumors, you'll see that telomerase has been activated one way or another, very complex, as I'll get into, but most advanced human cancers certainly show that telomerase is activated, and so it makes eminent sense that it's to maintain telomeres. But the balance can go in various directions. There's nothing that says that it just has to be completely on or completely off. As I said, there's multiple controls in the B cells, the germinal centers in adults. Hodes's group showed years ago telomerase is sort of balancing such that you get increased telomere length, but more likely in most cells in humans, the much more common situation is that you get some loss of the balance in the other direction. And so the idea would be if everything else were the same, which of course it won't always be, but if it were the same and you had a certain amount of telomerase, you might be able to stave off senescence by counteracting the shortening processes. But if you had less telomerase and just in the hypothetical way kept everything else the same, then you might get to the senescence quicker. So as I said, this is under a lot of different controls, and we could roughly classify them as genetic and non-genetic. And of course, in environmental, we would include things like infections, as you've heard, are so important in playing roles across the long life courses of people, and particularly in the decades and decades of human life. So all of these nuances are expected to interact. So how's it shaking out? Okay, so you can measure telomere length, you can do it quite reliably, and you can get a lot of answers both in disease situations and in normal populations. So measure telomere length, right? So this is the kind of thing that typically you would see. And I just grabbed this study. It's very typical. If you look across age, this is adults, what you'll see is two things. You'll see that there's a general decline, okay, and that shows up in a general way, although there's some interesting nuances for sure, but it only accounts for about less than 10% of the variation that you see in telomere length. What you're really seeing is this tremendous mess here, right? Now, molecular biologists hate this kind of thing, really loathe it, right? Because it'd be so, then it's a mess, right? So the question is there anything meaningful in this variation? Okay, so I want to give you one example, an unpublished example, which really makes the point, yes, you can start to extract information. So this is a very big study of 100,000 people done in California. It's the Kaiser Permanente. It's like an HMO sort of system. And so these are pretty diverse people. And you're looking at a sort of 40,000 feet up view. You're just looking at this swath of quite diverse humanity in California, as it happens. But we're not that weird. And so DNA was collected, and we measured telomere length. It happened to be salivary DNA, so it's quite a mixture of cells. You'd think this is really hopeless, right? But we measured telomere length in these 100,000 people and got it really reliably done. And then we waited. That's the time point. And then time went by. And then you can say, well, you can ask lots of questions because there was GWAS information and there's clinical information, but let's ask the simplest of all, who's dead, okay? So, right? And so I'm pointing out this is a wide age group here. You know, we're covering a big age group here. The median was about 60 or so years of age. The mean, sorry, about 60. But about 2,500 people had died in that time. So then you say, well, what relationship did that have? So it was really quite interesting because if you looked at people in the bottom quartile of telomere length, and if you set their odds of dying to 1, then everybody else was less likely to die by that amount. So what was particularly interesting is that we know from many, many studies, and we, meaning the field and many research groups around the world, have known that telomere length certainly correlates with a variety of different things, some of which are themselves risk factor for mortality. Obviously, age is one, sex is another one. You could correct for this. We also, in this cohort, had this kind of information as well as a lot more, but we could actually adjust for all of these, which again themselves are risk factors for mortality or not. But even after you adjust for them, it just says, it doesn't budge. So this says that this is independently a predictor for the next 3 years of whether you will live or die in a degree that is reflected in this odds here. So those are the facts, right? That's what you see. So that's a relationship. Now, no mortality causality is implied at all by this. This is just, they go together. Right, obviously the telomere shortness preceded the mortality, but doesn't say anything about causality. So, but there it is. So a similar relationship has been found, again, with multiple studies around the world of different kinds of cohorts and looking at all sorts of diseases. And they're the diseases that are really quite common in populations, particularly aging populations, and seeing quantitative relationships with shorter telomeres. Now, I won't even do credit to the enormous numbers of studies that have shown this kind of thing, but you see a pattern, and you see these sorts of things starting to emerge. Impaired immune function is measured in various ways. Some cancers, though some is very critical, I'll get to that more, lung disease, heart disease, diabetes, interestingly, vascular dementias, various kinds of osteoarthritis. So you see it, but again, no causality can be inferred from such sorts of things. So how do you infer, well, is there something going on that in which the shortened telomeres, which we know at the cellular level has clearly the kinds of effects that would certainly be predicted and reasonable to promote pathophysiologies, right? So, but this sort of thing doesn't tell you anything about it. And I just told you mortality risk. So how do you deal with causality? Well, the whole set of things we know are playing in to telomere length. There's certainly genetic components. There's variable degrees of heritability. Those degrees of heritability we showed in the 100,000 goes down and down with age, and that sort of implies, again, what other studies have correlated with, a number of non-genetic inputs into this. So we know there's going to be a great deal of complex inputs into this. And so what we're trying to look at is you look at this quantitative measure, you look at very clear readouts, and you try and infer causality. And of course, genetics is incredibly valuable for inferring causality, because I think most people in this room will think that genes do cause things, right? We sort of all pretty much agree on that. So let's have a look and see. And then, of course, humans, the clinic, the large population of, the 6 billion or so plus on the planet, provide more and more instances of what are, tragically rare, tragic and luckily rare diseases. And there are indeed people who have single Mendelian mutations that mean they only have half the amount of telomerase. And they're very clear in their molecular cause. We know exactly what goes wrong. It's a component of telomerase in most cases. So, for example, the telomerase RNA, or that was the first one discovered by Dokal and colleagues, and then HTERC mutations. And these other components are components of the telomerase complex. I told you there's more complexes. And these are known card-carrying, well-molecular cell biology studied components. We know what they do. Similarly, there are genes which we know are part of the telomerase maintenance, are part of the complexes on telomeres. So we know very molecularly well, perhaps not so well for RTEL, which is trickier, but a number of these we know what they're doing. We know what they're doing. And these mutations all can give very clear causality. They produce very short telomeres compared with the population at large, and indeed they have an impact. Now the impact is an interesting set because, of course, these are much earlier onset than in the population. These patients die earlier and earlier with genetic anticipation as they inherit shorter and shorter telomeres through the family, but they have an overlap with the kinds of diseases that we see in the common population. So the causality is very clear here, but it's more extreme because they've only got half the amount of telomerase compared with the population generally. But now, of course, that raises the question, well, what happens in the more nuanced situation in the rest of the population who don't suffer from such rare diseases? And so people have found that there's an anticipation of, I'll just show the Laureates et al. Study, the Bruneck study, which is shown here just for 10 years, and as they did initially, gone out to 15 years. They show exactly the same trends. If you measure telomere length just in the white blood cells, and then say who gets a cancer diagnosis, and just simply stratify it on who had the shortest or the mid or the longest tertiles of telomere length at this point, you can see it's a very clear graded prediction, and also of who died. And this is just all cancers lumped together. So now we're going to get more subtle. But this is the overall picture, right? Okay, so you can certainly say that there's an association. Now, didn't tell us about causality, though. This is what I just showed you. So now what about genetics? So now GWAS studies have started to add some nuance and some information here. So I'm going to summarize. I'll use Karl et al.'s very big GWAS meta-analysis publication, because this was very interesting. What they showed was that there was a set of genes associated with shorter telomere length or longer telomere length, depending on the common SNP. And they were in a number of the known telomerase genes, I'll tell you those, and they found that relationship was related to heart disease, and it accounted for some of the risk factor for heart disease. Not the majority, and that's a very important part, but it accounts for some of it. So what that starts to tell me is these genes, and I'm going to list them on the next slide. Okay, so we're looking at shortness. These genes, which are in this prominent group of GWAS associations, these ones, which we really know what they do, they are associated, they're SNPs, they're common SNPs, are associated, particularly in combination, with clear risk for, say, forms of cardiovascular disease. So we've got an element of causality there, and of course we expect it to interact a lot with other factors, but this is the kind of thing that's very, very useful. And they were able to define, Karl et al., a group of about six loci in which you could say this is a combination which will give you an increased risk of cardiovascular disease of a certain kind. So that's all I want to say about the causality in general for other diseases, but now, what about cancers? So of course, everything looks good, right? And then you get thrown a curveball. This is fascinating because here's what I just showed you. These could be associated with telomere length, but now this group and others now are showing that you could find SNPs in these known telomerase components and this known telomere influencing this requirement for telomere maintenance, RTEL1, and they found that there are SNPs that are associated with glioma risks. But these SNPs were associated with longer telomeres in the blood cells. So the opposite of what I just said was, the generality if you lump all cancers together. So just to emphasize the point, the idea is that since the telomeres are longer in blood cells, at least the simple inference, because it was related to TERT and TERC SNPs, because we know what those do, right? The inference is that there was more telomerase, and yet it was brain tumors that are affected in the other direction. So, as usual, cancer shows us its complexity of different kinds of diseases. It says, no, it's not just all one simple situation. So I'm just going to summarize a sort of bird's eye view of where I think it's all starting to look as if it's shaking out in terms of, again, being informed by genetics of various kinds. So a remarkable few papers came out in 2013, which were actually anticipated by a fascinating paper on chicken ALV insertional mutagenesis caused B lymphomas by Karen Beeman et al. In 2007. And it was like this. If you mutate the TERT promoter, and this happened very recurrently, initially shown in melanoma sporadic tumors, but also in a germline family studied here in Germany. And with this family, they had very high penetrance of melanoma. If they had a mutation in the promoter that just had this crummy little effect, when you put it under reporters, they found it just increases telomerase a little bit up. Wow, not much of a change, right? And this class now of tumors has been related to such changes. As I said, the melanoma family, the germline family, is very important because this was saying this is going on in a clearly causal fashion. And as I mentioned, it had even been anticipated in ALV-induced chicken lymphomas, where the most prevalent insertional site for these clonal lymphomas was actually in the TERT promoter, and it upregulated it just a little bit. So the important point is this is a subset. So just to say what I'd said before, if you have telomerase mutations which cause you to have only half the amount of telomerase, you only get a certain cancers, and I'll just list some of them here. These are the ones that you see they're most likely to be because the stem cell populations get exhausted as they in these very proliferative cell compartments, and then whatever the sequelae are, that you get cancers from these. So this is a subset of cancers that is really caused by this. The risk goes up about 1,000-fold in these individuals for some of these relatively rare cancers, but the causality is clear. But then I've just said, okay, well, actually, if telomerase, and I'm talking about TERT here, that's the TERT promoter, that's the one that's showing up over and over, little change in it that upregulates its action a little bit, its level a little bit, and there's another subset. Notice, by the way, the absence of breast cancers and prostate and cancers. This is some subsets of bladder cancer and some subsets of liver. But clearly, cancers are complex. So basically, the bottom line is that you don't want too much and you don't want too little. We're really teetering on a pretty dangerous spot because small changes could have effects that over the decades of life could have clear effects of problems. So I'm just going to illustrate that sort of quantitatively, like, mutation, giving you half the amount of telomerase. And we know this is about telomeres, but I'm going to suggest something else might be going on. Okay, your risk of cancer goes up. This is not necessarily, the dose relationship, but it's clear that from 1 down to half, that's pretty drastic for those individuals. Luckily, they're rare, but it's much, it's an undesirable situation compared with the wild type. But now you think, well, what if you just went up and up? Well, you get into a zone where it seems to be bad news again. Somehow, and again, don't take this too seriously in terms of quantitative, but at some point you reach a zone, as expressed this time in TERT expression, where again, you're having risks, again, for another set of cancers. So choose your poison. where you want to be is somewhere in this sort of Goldilocks zone. You know, Goldilocks said, I don't want the bed too hard, I don't want the bed too soft, I want it just right. You know, this is where it probably is the best place for humans to inhabit as regards risks of cancers. But as I said, the overall mathematics is that the telomere shortness in the blood cell is, you know, sort of down in this region, is associated more with incidence and mortality, as the Laureates et al. Publications indicate. But basically, we're in a pretty, you know, sort of precarious situation. So what this is saying is that it really would be good for us to learn more about what is regulating all of this, because it's obviously something whose exquisite regulation may be rather important. And this leads us back to something about cancer cells, which, as I said earlier, it sort of made sense that telomerase was very common in advanced malignant cancers. Well, you know, that makes sense. But actually, this is something a little bit odd, because telomeres are often really short in those cells. Why so much telomerase? I mean, they don't need that much to offset the various shortening processes. It's always been a bit of a mystery. So the question is something else going on? I'm going to say I think this is still an open question. There's been controversial things that have gone up and down in sort of fashion, and what I want to show you is just one set of things which we have been, you know, really led to by the cells themselves for TERT, and then again a set of things that we've found which we were led to by looking at cells in the situation of TERC, the telomerase RNA component. So, as I said, you know, lots of regulation can go on. Here's a TERT gene, here's an RNA gene. You know, the old idea was you'd have transcription, and then it's been known for a number of years that there's splicing variations that take place, but a full-length protein product from at least one of the splice variations, I'll show you there are more, will be able to assemble into an active enzyme combining with a telomerase RNA. You know, so that was the old the idea, but we knew there's a lot of complex controls. In fact, people have shown for a number of years that there's a plethora of spliced variants of the TERT protein. And some of them do something that looks really strange. They take out the reverse transcriptase domain, and they kill the ability of this protein to do any enzymatic activity whatsoever. So what's the use of this? So they'd always been dismissed as, yeah, they're sort of negative regulators and so on. Well, may not be quite like that. So I'll just show the two that are the most abundant. There's the full-length one, that's the one that has the full domain. And then there's something that's just got the historic name beta deletion. And that's a major form, as I'll show you, in cancer cells. It's also a major form in normal cells. And it makes a product that clearly can't do any reverse transcription. But it has domains here that can interact very well, we've shown, with the telomerase RNA. So this is totally capable of binding the telomerase RNA. This one is too, of course, but it can also do the enzymatic activity. So why bother with a thing like that? Well, if you look at a panel of breast cancer cells and you just look at the ratio of the different spliced variants, these are some minor ones, but what you can see right away is the two major ones, they're just shown as a percentage, not the absolute levels, which also vary a lot. You can see that they, you know, they're all over the place. They make a certain amount of sense, though, because if you divide breast cancers into basal versus luminal, for example, you can see that there's a sort of inverse relationship between the RT positive full length and the beta deletion RT null form. You know, this is up, whereas this is down in the luminals. The RT positive is down versus up in the basals. You know, so there's some regulatory class of things going on there. So it's not total nonsense. And in fact, when we quantify these, we can see a pretty clear kind of decision point that's made where, depending on the class of breast cancer, you'll either get, you know, this pathway versus this pathway. They're mutually exclusive going on. So we showed that the beta deletion is actually in normal cells, in cancer cells, this is leukemia cells. We showed that actually it does show up in polysomes. This is now the endogenous form. It's not an ectopic one. So it's made into a genuine protein. And curiously enough, if you tag it and express it, you can find it not only in the nucleus as you expect, but actually it's in the mitochondria. As has been long known for full-length TERT, completely mysterious what this is doing here, but it certainly localizes very specifically to mitochondria. And at least it has one effect we can discern in cells. And these are breast cancer cells, and it's long been known if you overexpress TERT, the protein, you can protect a variety of cells, cancer and normal, from apoptosis. So people always thought that's because you're beefing up the telomeres. So here's an apoptosis kind, which is the kind induced by cisplatin, and here's the control, and here's the one that got the extra HTERC full length. So no surprises there. That replicated what people had long seen. But the surprise was we could see the same thing by expressing instead equal amount of the RT null one. And this just shows a couple more cell lines, exactly the same kind of thing. So the curious thing is that, here's what you'd think would be the most useless protein in the world, right? But cancer cells do express it often more than the TERT, which does reverse transcription. And it can actually inhibit, at least we showed very clearly here, this sort of apoptosis which is cisplatin induced. So, not irrelevant to a clinical situation. And I didn't go into all of the different controls. It's under a lot of different controls, as is the full-length TERT. And I just mentioned the cancer subtype-specific alternate splicing as just one example. Okay, so in other words, when you have a mutation in a TERT promoter, as is so common for a subset of the cancers, the brain tumors, the sporadic melanomas, now subsets of liver and subsets of bladder, just as examples, you're actually increasing this as well. So, we've got to understand the biology of this much better. So that's the first example of, why we think that there's more to be asked. Now, what about TERC? Okay, so here's what we know. We know telomere maintenance, you get splicing, decision made, this one will maintain telomeres, this one won't. And, so that's the old familiar part of telomerase. And now we say these, can act anti-apoptotically. This was known for the full length. We can now add that. Okay. Now, what about TERC? So TERC has always been the lonely sort of orphan. And yet there's something funny here that's always been just glossed over. It's expressed very widely. So, for example, fibroblasts, which have essentially no telomerase, no TERT expression, you get nice expression of TERC in there. What's going on? It's always been just one of those things that was there and no one really had a hypothesis. But we came across what we think might be an interesting possibility. So, as I said, the known action of TERC, of course, is to assemble into telomerase. This is already known, right? So we have this set of known things. We knew this. And now what I want to show you is just very briefly a bit of the experiments. We just published them recently on not just this, but we think that this might be going on. That when we have now a bunch of experimental data that said that this component actually protects apoptosis in stimulated CD4 T cells. That's the system that we're using. And we're looking very short-term experiments, just over days. And so now we think that this is another thing that TERC is doing. Okay, and it's not doing it through assembly into an active telomerase. So let's just go through just a bit of the data because I don't want to spend too much time. Now, the beautiful thing about all the studies in humans is that there have been point mutations found in unfortunate individuals who've inherited a mutation, and they're very well characterized molecularly. So here's the telomerase RNA. Only this part's the template here, and making the template for synthesis of telomeric DNA. There's all this other part. And of course, some of it's to do with HTERC binding. Some of it helps the enzyme RNP do its polymerization. And then there's another big domain here, which is involved in stabilization. And this is actually a telomerase. So these parts are to do, particularly here, with, and this part with binding of TERT and enzymatic, the traditional familiar parts. This part stabilizes the telomerase RNA, and this actually, it's borrowed a box H ACA domain from that particular class of small nuclear RNAs. So people have thought, well, this is stabilization of this, makes perfect sense, and that's with a complex of known proteins. One of the mutations is in this absolutely essential region for telomerase activity, and it's a point mutation, and it kills the enzyme dead as a doornail for enzymatic activity, but it's perfectly stable. Okay, so dead. And another one I'll talk about, again, comes from human disease mutation, a couple of base pair changes, a little deletion, a tiny bit in this absolutely essential pseudo-knot, dead as a doornail for enzymatic activity, absolutely dead. So imagine our surprise when we did the following experiment. So we have in cells, we actually have telomerase, HTERC is assembled into an enzyme and some of it's not. Actually, the HTR is also some assembled into an enzyme and some is not in CD4 cells, fresh cells from healthy young donors. So if you just look at telomerase activity, you've put in a control scrambled vector. In this case, you get a certain amount of telomerase activity, and then you treat them with dexamethasone, long been known as a mimic of the normal stress hormone, to cortisol to increase apoptosis. So there's a dexamethasone-induced level of apoptosis. So then we put in we overexpress the wild type. Telomerase activity, of course, goes up. Made perfect sense. No effect on the dex apoptosis. It stayed just as bad, right? But then we overexpressed at the same level this point mutation, catalytically dead. Telomerase activity, of course, didn't go up. And now, though, we got protection from this DEX-induced apoptosis. We got much, much less. So just that change here. So this was weird, right? So then we did it with the other point mutation, got exactly the same result. So we said, well, what's here? Do we need this region in here? And so we switched out the BOX-H-ACA domain with another SNO RNase, and that one wasn't able to protect from DEX-induced apoptosis. So this is just that graph shown bigger, just to make the point that here's the one that does protect, and here's the one that lacks the 3' portion, even though it's got a perfectly good SNO domain, it's perfectly stable, but it can't do this. So there's something interesting down in that region, and that's what we're trying to trace molecularly down. So we don't have the answer. But I just want to show you that the other way around, where you said, well, now if you reduce the amount of telomerase RNA, because you always worry you overexpress something weird happened. So we reduce the telomerase RNA. Now this is what happens in culture with CD4 cells. If you stimulate them and then you, oops, I'm going excitingly backwards. Okay, if you stimulate them and then let them go through their normal course of proliferation, there's a sort of tracking of telomerase activity, goes up and goes down, and then you can re-stimulate them and they go up and down. So this is just in a period of a few, couple of weeks in this experiment, a few weeks. So basically what we did was we said, well, let's reduce the telomerase components. So we did that, and you can nicely reduce the telomerase RNA. This is quite a lot of work to do this, actually. But what we got was the cells really hated it, right? So the number actually went down upon re-stimulation. So this was saying it depended upon at least this component. So let's look at this time point and quantify it. So this is the kind of thing I just showed you. Use the controls, and here is the telomerase activity at that 3-day time point. And the live cell count went down. Now we reduce the telomerase activity level to the same degree by knocking down TERT. See, it's the same degree. The cells are just fine. So we knocked it down to the same degree, and yet we did not affect the cell count. And in fact, what it's looking like is it's an apoptotic response. So here, for example, is the caspase 3-7 activity assays. Okay, so we then, of course, went through the whole rigmarole of looking at the telomeres. And I will just say all the results we found. We looked at every time point through these time courses, looked at telomere length, damage, distribution of lengths, everything we could think of. Nothing was different. This is a short time frame, by the way. So it's nothing to do with telomeres as far as we can tell. And this really sort of reinforces that point. So there's other data, but essentially that's the finding, that we find that it's inducing, it's this pathway, it seems, as judged by looking at, it's caspase 9 induced, etc. There's BIM involvement and so on, maybe a little PUMA involvement. We don't seem to be getting the death receptor pathway, so it looks like something is going on in this pathway. But we don't know why. All we can say is that this is happening. So we have the situation, which was old, we'd seen before, the anti-apoptotic effects of TERT in the setting of cancer cells, and now we're in really normal cells. And very strangely, we're seeing this apoptotic response. And by the way, we can shift we think this is alternative pathways. Why? Because we can push the HTERC into this pathway, for example. Oh, so we can take TERT down and push it into this pathway, and we get better anti-apoptotic effect. So this is strange. You put TERT down. This is short-term experiments, and it actually is more anti-apoptotic. And this is different forms of, different settings of apoptosis, by the way. And then conversely, we can push them into this pathway by expressing more TERT, and we find that now there's less of that protective effect. I won't show you all those results, but it's looking as though TERC is going into different pathways, and this is the old familiar one, but now we have a whole new kid on the block. We don't know what this means at all, so I've just thrown out a couple of curious possibilities. Maybe the RNA forms a different conformation. It's known to form different conformations during the enzymatic cycle of polymerization in telomerase RNP. So it's quite feasible that it is able to form some alternate conformation. Or maybe it's processed. The obvious candidate is to ask if it's processed into a mere. And a really interesting hint is that certain SNO RNAs are known to be processed into meres. So that maybe that's what's happening. There's actually a microRNA that TERC is producing. We don't know this for sure. We don't know if it's the whole entity or this, but we do know it needs the 3' portion. So anyway, the point of all of this is to convince you that we thought we knew quite a lot about telomere maintenance and the machinery, and there's been a whole part that we really don't know. The HTERT, non-reverse transcriptase competent beta minus, very prominent in, as I said, normal cells in certain settings as well as in cancer cells, and an HTERC lurking around in all sorts of cells that don't have telomerase. And here's something we've uncovered, which I think is going to be very provocative to try and understand more about what's going on there. So, in summary, if we put it back in the context of cancers, it's pretty clear that a small perturbation of TERT, as I said, has these wonderfully interesting and unexpected effects that came from doing cancer genetics, cancer genomics, molecular studies, and saying that it really is oncogenic for certain subclasses of cancers. Now, I think it's very, very reasonable it's doing it through better telomere maintenance. I really think that's probably a primary thing, but we shouldn't neglect maybe this is also playing some kind of a role. And then the second unexpected thing is that well, from the genetics, first of all, we knew having half as much TERC was a big problem, but curiously enough, although it's probably very important in the cancer cells, and quite possibly important in immune cells, which will control certain cancers too, and this has been much less well thought of in terms of the phenotypes of the HTERC cancer patients, but I think this is something one really should think about. And then another question would be, if you've got only half the amount of this, which is the case, then that's going to have real functional impact on immune cells, these mature cells that we're talking about, at least via that one pathway that we've been able to uncover in CD4 cells. So I think what we have is a set of things where the genetics has really been very informative. The cell biology is telling us things that rather small changes on these components could have impacts on as well. And just to sort of round out the picture, we now know that there are a lot of non-genetic influences on telomere maintenance in humans. And Petra alluded to some of the mini studies that we and others have been involved in. And so all of that is going to play in with the courses of these diseases that we can see new aspects of here. So I'm going to say thank you very much. And again, congratulations and best wishes for another 50 years.